Hi, my name is Dejesus. I'm a bio major here at Keene State College. This is my poster project for the bioinformatics class, Bio 490. So I originally wanted to have a sort of medical relevance to my poster, as I do want to go to PA school eventually after graduation. So I heard about bacteria that were being used as vectors for anti-cancer therapy treatments. So I stumbled across this bacterium. It was actually suggested by Dr. Launen in class. This bacteria, Listeria monocytogenes, which is a foodborne pathogen, and it causes listeriosis. Some of the symptoms really quick, typical of most foodborne pathogens: fever, diarrhea, vomiting, nausea, that kind of thing. But super severe infections, if the bacteria actually leaves the gastrointestinal tract, can cause spontaneous miscarriage. Typically affects those with weakened immune systems and pregnant women and those who are very young. Mostly found in untreated or poorly treated dairy foods or unprocessed meats. So, from a bioinformatics approach, this is the pipeline outlining the basic sort of stepwise manner in which we analyze the genome of the bacteria. It starts here when we acquire the DNA through raw samples, which after a series of steps through sequencing, through Illumina sequencing, we have these various programs that sort of treat the raw data, which is basically just giant files called contigs of nucleotides, like raw DNA sequence. And through running these different programs, we get these different outputs, which we can use to analyze the genomic data, sort of like this, which measures the contig size of certain strains versus a reference strain that you can get offline. These are the 8 strains that I wanted to kind of study to see if they were potential cancer treatment strains. Sorry, if they were potential, what am I trying to say? Potential, I don't know what to say.
Vectors.
Potential candidates for cancer treatment therapies. Basically, the way that Listeria monocytogenes is used as a cancer treatment vector is through this gene here, called the HLY gene, which codes for a protein called Listeria lysine O. Basically, this bacteria acts as sort of like a parasite, where after it sort of infects this host cell, it is engulfed into a vacuole inside of that cell. But unlike most bacteria, or some bacteria, sorry, using this protein, it has the ability to kind of bore out of that vacuole and escape into the cytoplasm of the cell, where it lives, reproduces, and then eventually moves on and infects another cell. The reason that this is great for cancer treatment therapy is that companies can take sort of hybrids or altered versions of this bacteria and attach antigens, which is what the body uses to recognize foreign bodies and attack them using an immune response. You can attach an antigen to this protein that it uses to break out of that vacuole and attach it to the outside of a tumor cell, which then triggers an immune response to actually attack and inhibit tumor growth. It doesn't necessarily, destroy the tumor, but it does sort of restrict further growth of that tumor. Basically, out of all the strains that I analyzed using this sort of pipeline, all of them were positive for the HLY, or the Listeria lysine O gene, except for this strain right here. There could be a lot of reasons why it doesn't have that gene, whether it be a new strain, or it could be a strain that was used for maybe another experiment or for another purpose. These other genes are sort of other virulence genes that code for things like cell wall formation for bacteria, or these Act A allows the bacteria to move within the cytoplasm of its host cell. But basically, that's sort of the general explanation as to why it's so important for medicine, and why using bacteria, which are usually thought of as bad for us, can actually be used in a positive manner. So, and then there's just the crystal structure of the Listeria lysine O gene. So, yeah, that's it.
Very good. Thank you.